Okay, this is chapter 21 and it addresses the immune system. And we're going to discuss all the components of the immune system, how it protects us against disease, and where the important structures are located. And we'll talk about the different types of defense, including um, innate immunity, acquired immunity, and um, the use of what we call our barrier functions in order to keep us healthy most of the time. Um, we'll also talk about where the immune system comes from and how it gets established and how it determines whether or not something is a threat to the body or not. We have different systems that are designed to protect us against disease because we live in a world that's filled with stuff that tries to make us sick, and these things are called pathogens. Okay, pathogens can be living or non-living. Living pathogens include bacteria, mold, um, or you could say fungus, parasites, all right, um, even in some cases insects, right, or protozoa in some cases. Now, some pathogens are not living, and those include viruses and toxins. Okay, but the point is, they're they're around us, they're in us, they're on us all the time. And if we didn't have a way to defend ourselves against disease, we would constantly be sick. So when we talk about non-specific immunity, we're talking about a body system that uses the same approach regardless of the nature of the pathogen. Our first line of defense includes our mechanical and our chemical barriers. Now, what are those? Okay, um, our our mechanical barriers are our skin and our mucous membranes. Okay, and the presence of things like the the hairs in the nose, the vibrissae, that act as a filtering mechanism. Um, the chemical barriers are the secretions that come from those mechanical barriers, and they include mucus from the mucous membranes, um, oil and sweat that comes from the skin. Okay, and there are components uh, in those secretions that retard bacterial growth. Okay, there are antimicrobial compounds, and there's also an acidic pH that's produced on the skin, especially, as well as a high salt content that tend to retard growth. And the fact that the upper layers of the skin are constantly shed and remade also cuts down on the ability of the skin to be colonized. Okay, our tears, our saliva, our sweat also contain things like lysozyme, which weakens bacterial cell walls. Um, Meganins are components that are found in ocular secretions, and they punch holes in bacterial membranes and make it easier for them to basically be killed when they have an osmotic shock and causes them to rupture. Okay, but all of this is going to use the same approach regardless of the identity of the pathogen. The next line of defense in the event that our mechanical barriers are breached includes phagocytosis, which is the ability of certain cells in the immune system to eat pathogens through essentially consuming them and forming a little vesicle around the pathogen, and then inside the cell, lysosomes fuse with that vesicle and break up whatever's inside it, and that destroys it. Okay? The inflammatory process, which we'll learn a little bit about in a minute, which is essentially where an infected or an injured area turns red, swells up, warms up, and hurts. Okay, that's designed to bring blood to the area, and with the blood come the white blood cells, and that helps protect against disease. Fever, which creates conditions that pathogens don't like, but which our white blood cells do, and it makes it easier for us to kill them. Okay? There are proteins that are going to defend against our pathogens as well. These are contained primarily in the vesicles of granulocytes, which are basophils, neutrophils, and eosinophils, and they essentially either punch holes in membranes of pathogens and cause their cytoplasm to leak out and they die, or they induce cellular suicide called apoptosis. Okay? And then natural killer cells that basically punch holes in either infected cells or cancer cells and cause the cytoplasm to leak out and they die. But in every case, these chemical weapons are not specific for the pathogen. They are going to use the same approach regardless of whether it's a bacteria or a parasite or what have you. Okay. The third line of defense is different. The third line of defense is based on the presence of molecules on the surface of the invader called antigens, which are specific for that invader. And we generate then a team of white blood cells called lymphocytes that are only going to go after whatever the antigen is attached to. Okay, if it's attached to an infected cell, it's going to kill the infected cell. If it's attached to the pathogen, it's going to try to kill the pathogen. And there's a variety of techniques that it uses to do that. Okay, so you can see here all the different components of immunity that keep us relatively safe all the time. The digestive system is kind of interesting. If we look at the back of the throat, we have tonsils, which are basically collections of lymphocytes and macrophages, and they trap ingested pathogens. Okay, the mucous membrane that lines the entire digestive system from the end of the esophagus to the beginning of the rectum is going to produce mucus that flushes pathogens out and traps them and keeps the pathogens from penetrating through and getting into the blood. And then the fact that the stomach is an acid bath that sterilizes the food that we eat is another way that we again cut down on infection. And we should also point out that in the layer of tissue under the epithelium, the mucous membrane that lines the digestive system, are lots and lots of white blood cells that are going to defend against anything that gets through the epithelial layer and tries to get into the bloodstream. Okay, so that's just an example of how really all of these systems are present in the digestive system. The point being that again, you need all of these acting together in order to keep us from getting sicker quicker. Okay, so let's look at phagocytosis, right? Essentially what happens when you injure or infect tissue is that there are chemical signals given off that draw white blood cells to that area, and then the white blood cells are going to start by eating pathogens and cellular debris. Okay, so let's take a look at how this happens.
In phagocytosis, phagocytes are attracted to the area of invasion by chemical products of the microorganism, phospholipids released by injured mammalian cells, or by components of the complement system. The phagocyte moves into the area of invasion and then attaches to the microorganism. The attachment is mediated by a variety of surface receptors, including antibody, lipopolysaccharide, and complement receptors. For example, the C3b component of complement coats bacteria or other particles and then binds to C3b receptors on the phagocyte. This process of coating to enhance phagocytosis is called opsonization. The microorganism is then engulfed by the phagocyte into a vacuole known as a phagosome. Vesicles in the cytoplasm called lysosomes fuse with the phagosome, releasing digestive enzymes such as lysozyme and proteases into the phagosome. The structure resulting from this fusion is called a phagolysosome. Inside the phagolysosome, microorganisms are killed and digested. Finally, the digested contents of the phagolysosome are eliminated from the phagocyte by exocytosis.
The inflammatory response is an important nonspecific defense against tissue damage. It begins when injured tissue cells release chemical signals that activate the endothelial cells of nearby capillaries. Within the capillaries, adhesion molecules called selectins are displayed on the activated endothelial cells. These adhesion molecules attract neutrophils, slow them down, and cause the neutrophils to roll along the endothelium. As the neutrophils roll along the endothelium, they encounter chemicals that activate integrins, which are adhesion receptors on their surfaces. These integrins then tightly attach to adhesion receptor molecules on the endothelial cells. This causes the neutrophils to stick to the endothelium and stop rolling. This accumulation of neutrophils along the walls of the capillary is referred to as margination. The inflammatory mediators released by the injured tissue bring about changes in the environment that cause mast cells to degranulate and release histamine. Histamine causes vasodilation and an opening of the junctions between the endothelial cells, allowing fluid and leukocytes to leave the capillary and enter the infected tissue. The neutrophils now undergo dramatic changes in shape and squeeze through the endothelial wall into the interstitial tissue fluid. This process is called extravasation. The neutrophils, followed by other types of phagocytes, are attracted to the damaged site by chemotactic substances released by bacteria and tissue breakdown products. They ingest and destroy invading bacteria.
Hey guys, if you can't tell by my voice, I've been pretty sick this week, but I was determined to get a video out to you guys nonetheless, and I figured it would only be suitable to answer the question, why do we get fevers? So, let's dive into it. A fever can simply be anything different than the average 98.6 degrees that the body is normally at, but since the body can fluctuate a degree or two during the day due to environmental factors, that isn't a very accurate definition. A better way to characterize a fever is by oral temperatures higher than 100 degrees Fahrenheit, but since oral temperatures still aren't incredibly accurate, the best way to measure a temperature is to go through the ear or rectum, and a fever then would clock in at temperatures at 101 degrees Fahrenheit or higher. Fevers are of course most commonly caused by viruses, but they can of course be caused by other things such as toxins in the environment, a messed up hypothalamus, or of course a heat stroke. But as the number one cause of fever today is a virus, that's what we'll be focusing on today. When a virus infects your body, it oftentimes travels through your bloodstream. But thankfully for us, our body has white blood cells. These guys travel around our body, and when they find a harmful bacteria, they produce pyrogens that travel to our brain. These pyrogens then travel to the hypothalamus area in our brain, the area that regulates our body temperature. Our hypothalamus in turn then cranks up our body temperature and gives us the fever. But what's the purpose of making our body feel miserable? Well, most bacteria that lives in our body lives at the nice and cozy 98.6 degrees Fahrenheit that our body normally functions at. So, when the body heats up, the bacteria either stops reproducing or dies, making our body get better over time. But with this information in mind, a controversial topic is brought up. Should we take medication to lower fevers? The answer is no unless it's a dire situation, because by lowering body temperatures, you're allowing the infection to continue to grow and hurt us. And let's not forget about those nasty side effects. Some side effects of ibuprofen, the active ingredient in most fever-reducing medicines, are, wait for it, chest pains, weakness, upset stomach, diarrhea, rash, dizziness, headache, pain, numbness, bloody stools, shortness of breath, slurred speech, seizures, itching, and chills. So, next time you're taking a sick day off of work, put that medicine away and let your body do its work. Disclaimer: the statements and ideas provided here in this video have not been approved by the FDA, the Food and Drug Administration. These statements are not intended to diagnose, treat, cure, or prevent any disease, so if you're having any problems with your body, be sure to visit a professional healthcare advisor. Thanks for watching. If you enjoyed, be sure to rate, comment, and subscribe. Like I said, this week I've been pretty sick, so sorry for the kind of sick voice and the shorter video, but hopefully next week will be a much better video. Once again, thanks for watching, and if you would like to see some other content by Science Saturdays, be sure to click on the pictures below. Science Saturdays, bringing free education to the modern world.
When a cell is infected by a virus, the virus enters the cell and produces structures that are not found in uninfected cells. The presence of this viral material signals the cell to produce interferon. The interferon moves out of the cell and attaches to receptors on nearby cells of the same type. The cell that produces the interferon is unable to save itself. The virus replicates in this cell and then moves out to infect nearby cells. The nearby cell that already has interferon bound to its surface responds in several ways, including production of enzymes that degrade messenger RNA and prevent protein synthesis. Thus, a virus can attach and enter the cell, but completion of the viral replication cycle is prevented.
Regardless of whether the complement system is activated by the classical, alternative, or lectin pathway, the activated complement proteins are involved in several important processes associated with host defenses, including inflammation, attraction of neutrophils, opsonization, and cell lysis. Complement C3A participates in inflammation by inducing changes that cause the walls of local blood vessels to become more permeable. This facilitates movement of phagocytic cells from the circulation into the tissues. Complement 5A induces a directed chemotactic migration of phagocytes to the site of complement activation. Complement C3b functions as an opsonin. It binds to microbial cells, making them more susceptible to phagocytosis. Complement proteins C5b, C6, C7, C8, and C9 react together to form a hole in the cell membrane. When C5 convertase converts C5 into C5a and C5b, the C5b binds to the cell surface and combines with C6. C5b6 binds C7, enabling it to insert into the membrane. Binding of C8 is followed by binding of several C9s. The hole generated by formation of this membrane attack complex causes the cell to lyse.
Okay, so if we look at another component of the second line of defense, natural killer cells are lymphocytes that destroy microbes and, more importantly, go after cancer cells. A cancer cell is just a cell that is mutated and begins to divide in an out-of-control fashion that is eventually going to produce a cluster of cells that can remove nutrients and oxygen from the blood at the expense of normal tissue, and even invade and destroy normal tissue to use in order to get space to grow and to really break down those normal tissues to even use them as fuel. Okay? Eventually what happens is that so much space and nutrients is removed from healthy tissue that it dies, and as tissues die, organs die, and as organs die, organ systems shut down, and as organ systems shut down, eventually the organism, you, are going to die. And so this is the body's normal way of trying to knock out cancer cells, in addition to the use of something that's called interferon, which is a secretion, a molecule that warns neighboring cells of the presence of viruses, which can cause cancer, as well as the production of a cancer cell. Okay, And essentially, the reason that this is produced in only um, localized regions is because in large amounts, interferon is actually toxic to the liver. Okay, so the body uses this, but it uses it sparingly. Okay, so our next topic is looking at specific immunity.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
Okay, we looked at specific immunity, and what we need to understand about an antigen is that it's a molecule or collection of molecules that identifies an invading organism to the immune system um, as not belonging to the body. Okay, so what the immune system does is it distinguishes what we call self from non-self. Okay, an antigen can be really any class of molecule. It could be a glycoprotein, it could be a protein, it could be a lipoprotein, or any combination of any of these. Okay, and as long as we have the ability to produce a T cell that recognizes this collection of molecules, then we can generate a team of T cells that can go out and kill anything that that antigen is attached to. Okay, the B cells assist by producing antibodies that bind to that antigen as well, and the result of that is that we have now a two-pronged attack. The antibodies are designed to go after pathogens that are out in the open by attaching to them and tagging them for destruction, while the T lymphocyte's job is to show up at infected tissue and destroy it. Okay, and so T lymphocytes come in different forms. Okay, there are cytotoxic T cells that do the work of killing infected tissue. There are helper T cells that do the work of finishing the job of activating cytotoxic T cells and B lymphocytes. And then there are regulatory T cells that turn off the entire process once the immune system has won the battle. Okay? T cell activation is a multi-step process. Basically, a macrophage will eat an invader and chop it into little bits and display those little bits on its surface along with what we call a major histocompatibility protein. And in a macrophage that is an antigen presenting cell, that's called an MHC2. Okay, and then what will happen is that the helper T's that recognize that collection of molecules by binding to it are going to be turned on. Okay, and they will then interact directly with B cells that recognize the same antigen and turn them on, and they will also indirectly activate cytotoxic T cells by secreting chemicals that turn them on. Okay, so the helper T is a pretty important population of cells. Without them, we wouldn't get an effective third line of immunity in order to keep us disease free. And this is a problem in a disease known as AIDS, where the helper T cell population is targeted by the AIDS virus. The result is that we don't have an effective immune response against pathogens that leak through our first and second line of defense, and the result is that we succumb to cancers and infections that normally are not lethal. Okay, so this is a look at T cell dependent antigens.
Antigens are processed differently, depending on whether they originate within or outside the cell. Proteins produced within the cell, such as viruses or self-proteins, are broken down into fragments. Fragments of foreign proteins are antigens. The antigens are then transported into the rough endoplasmic reticulum. The antigens combine with class I MHC molecules. The combination is then transported to the Golgi apparatus and from there to the plasma membrane. Foreign antigens presented on class I MHC molecules stimulate cell destruction by activated T cells. Self-antigens are not recognized by the T cells and do not stimulate cell destruction. A different process occurs if the antigen originates outside the cell. Phagocytes, such as macrophages, ingest foreign particles such as viruses and bacteria by endocytosis. The foreign particles are broken down into fragments within a vesicle. The vesicle containing the foreign fragments fuses with vesicles from the Golgi apparatus containing the class II MHCs, and the two structures combine. The MHC class II antigen complex is transported to the plasma membrane. The displayed MHC class II antigen complex can stimulate other immune system cells to respond to the antigen.
Proteins generally require the cooperation of T helper cells, Th2, to stimulate B cells into becoming antibody-producing cells and memory cells. Such antigens are therefore said to be T-cell dependent. The protein antigen is first ingested and then broken into peptide fragments by an antigen-presenting cell, such as a macrophage. The antigen is then moved to the surface of the macrophage on a class II MHC. The T-cell receptor on the surface of a T helper cell recognizes the peptide antigen that is being presented on a class II MHC of the macrophage, and the T helper cell becomes activated. The activated Th2 cell is now capable of stimulating B cells. B cells also ingest antigens. The antigen reacts with an antibody on the surface of the B cell and is then internalized, digested, and presented at the surface of the B cell on a class II MHC. The antigen class II MHC complex of the B cell interacts with the T cell receptor on the activated T helper Th2 cell. The T helper cell produces cytokines, which stimulate the B cell to proliferate and differentiate into antibody-producing plasma cells and memory cells.
The immune system can produce antibodies against millions of different antigens. B cells, which are constantly produced from lymphoid stem cells, are found in the blood and lymphoid tissues. Each B cell can synthesize only one of the millions of possible antibodies and displays this antibody on its surface. When an antigen meets a B cell having a surface antibody of the proper specificity, it complexes with the antibody. B cells that are not activated do not develop further. Stimulation of only the B cells that carry antibodies that react with antigen is referred to as clonal selection. Following antigen-antibody interaction, antigen-antibody complexes aggregate on the surface of the cell. This is called capping. The antigen-antibody complexes are then internalized. The activated B cell swells and begins to divide rapidly, producing a B cell clone. The B cells then differentiate into plasma cells and memory cells. Plasma cells produce the specific antibody that provoked its formation, whereas memory cells remain in circulation but do not produce antibodies. Memory cells can become activated by a later challenge by the same antigen. Many antibody responses require signals from T helper cells. The T helper cell is stimulated by an interaction with an antigen-presenting cell such as a macrophage. The macrophage ingests the antigen, digests it, and presents it on the cell surface via a class II MHC. The macrophage then activates a T helper cell that carries a T cell receptor capable of recognizing the antigen on the class II MHC of the macrophage. The B cell presents the antigen on its surface using class II MHC. The activated T helper cell interacts with the B cell and secretes chemical signals called interleukins to stimulate the B cell to differentiate into a plasma cell and produce antibodies.
Okay, one of the things that we need to appreciate about antibodies is that they come in different classes, okay? All antibodies have the ability to bind to an antigen that's on the presence of a pathogen and target it for destruction, but different antibodies in different locations are capable of aiding the immune function in different body compartments, okay? These immunoglobulins attach to the invader, and the different classes are IgG, IgA, IgM, and IgE. They're found in different locations on the body, and they all act to target antigen attached to pathogen for destruction by the other components of the immune system. Okay, so let's see exactly how this works.
Antibodies are proteins that play an integral role in the complex system of specific immunity. They bind to and inactivate foreign proteins called antigens. Each type of antibody binds to one kind of antigen molecule. B cells are one of the two types of white blood cells that make antibodies. When a bacterial cell enters the bloodstream, proteins on its surface act as antigens and trigger response by the immune system. These antigens will bind to the unique B cells that have the corresponding antibodies on their surfaces. These B cells now divide. Some continue to divide, producing many plasma cells. Plasma cells secrete large quantities of antibodies into the bloodstream. This division of only the appropriate B cells is called clonal selection.
So, when we look at these different antibody classes, what we find with IgG is that it's generally located in plasma and body fluids and is effective against bacteria, viruses, and toxins. And this is really the second antibody to show up after an initial infection. Okay. IgA is found in breast milk, tears, and gastric juice and protects against infection there and is one of the agents of what we call natural passive immunity. Okay. IgM is found in plasma and is associated with blood type compatibility or incompatibility. It also is one of the first antibodies to show up and fight bacteria, viruses, and toxins when we're initially infected, right? So the IgM actually comes in first and the IgG comes in the blood second. IgE is involved in hypersensitivity reactions and is an agent of a condition known as anaphylactic shock when it essentially malfunctions. Okay? So one of the things we have to appreciate about the immune system is that when we're exposed to an antigen initially, the primary response is the first attempt to defend against infection on the part of the lymphocytes, okay, from our third line of defense. And the first antibodies to show up to do that are the IgM class, okay. They bind to the antigen and target it for destruction. They have the ability to attach to a series of blood proteins called complement, which destroys whatever the antibody is attached to. Okay, so it's one of the ways that we complement the action of antibodies in fighting disease. And these proteins are always around in the bloodstream, and they're made by the liver. Okay, and then the secondary response comes in, and this is when you see the same the same antigen a second time, okay? And the result is that we generate a greater number of antibodies more quickly, and the result is that we're able to knock the pathogen out before it gets a chance to make us sick. So this is what immunity is all about, right? We get an initial exposure and we knock it back using our own tools, our own white blood cells. Then we generate memory cells for the lymphocyte team that did that, and the next time that pathogen shows up, we're able to much more effectively knock it down. Okay. In both of these exposures, we have the IgM class coming up first and the IgG class coming up second during the exposure. The same thing happens in the secondary response, but it happens more rapidly. Okay, and the result then is that we more effectively knock out the pathogen. And this is one of the reasons why frequently, if you were extremely sick as a child, but were able to recover from childhood diseases using the immune system, you tend to be relatively healthy as an adult because of this phenomenon known as memory. Okay? And these memory cells stay with you basically your whole life. So, immunity comes ultimately from our DNA. Okay, when we're making the third line of defense, need the ability to recognize a large variety of antigens. So the way that we do that is that we take genetic material responsible for producing the antibodies and the T cell receptors that do that job, and we shuffle and combine them during the formation of the immune system, while we're still inside our mother's womb, to make billions of different, what we call them, specificities, right? So these would be antigens of different types that these antibodies and T cell receptors can attach to, okay? And so you end up with between a billion and a trillion different specificities for your T and B lymphocytes, and that is not going to include your own antigens, right? Because we kill those those lymphocytes that would attack and destroy our own tissue by either killing them outright or by rendering them in a state of what we call anergy, where they're alive but they're not participating in the immune system. Okay? And so at the end of the day, we end up with a population of white blood cells that can tolerate our own antigens but destroy antigens that come in from the outside. Okay? Now, when we talk about immunity, we're talking about the ability to produce a team of white blood cells or to utilize the products of white blood cells in a way that defends against disease. Naturally acquired immunity is what happens as a result of our activities of daily living, okay? While artificially acquired immunity happens as a result of a deliberate exposure, okay? Or a deliberate injection of a product of a team of lymphocytes, okay? The way that this breaks down is if we talk about natural passive immunity, that would be an injection of antibody, that would be acquisition of antibody through normal activity, okay? So we get that in mother's breast milk, and we get that while we're in our mother's womb through the blood supply, okay? And those antibodies are prophylactic. They're temporary protection, okay? If we're talking about artificial passive immunity, then that's an injection of antibody from another source that provides temporary protection for a short period of time, okay? Active immunity is different, okay? In active immunity, we either naturally acquire that by getting an infection and knocking it down, and in the course of that making memory cells, okay? Artificial active immunity is where we get a vaccine that induces the production of a team of lymphocytes that will recognize, bind to, and destroy the antigen wherever it's found, okay? So the active immunity is always a better deal because that stays with you your whole life, while the passive immunity is more of a temporary state, okay? So that's an example of naturally acquired, right? Active and passive, right? Artificially acquired, right? Active and passive. Right? Allergic reactions are essentially inappropriate responses of the immune system to something that's not actually a threat, okay? And the result is that the effects of the immune action are actually what make us sick, okay? So it comes in different types, right? In anaphylaxis, okay, we have an allergen activating a B cell. The activated B cell forms a clone of antibodies secreting plasma cells, which secrete large amounts of IgE against that allergen. The IgE binds mast cells and they release histamine, which promotes inflammation. And then the result is that we end up with inflammation over a large area, okay? We can take a look at some of the different types of hypersensitivities here.
Some people develop an allergic reaction or hypersensitivity when exposed to substances such as dust, pollens, animal dander, or penicillin. Sensitization occurs when the antigen makes contact with some part of the body. The antigen is taken up, processed by antigen-presenting cells, and presented to T helper cells. Tissues under the mucous membranes are rich in B cells committed to IgE production, and IgE-producing cells are more abundant in persons susceptible to allergies. The T helper cells produce cytokines, which stimulate B cells in the area to proliferate and differentiate into IgE-producing plasma cells. As IgE is produced in specific areas of the body, the IgE molecules attach to nearby mast cells. The individual is now sensitized to the antigen. When exposed to the antigen for a second time, the antigen immediately binds to the IgE antibodies, which are already attached to the mast cell. Within seconds of this binding, the mast cell releases histamine and other chemicals that cause an inflammatory response. These chemicals trigger a variety of symptoms such as capillary dilation, airway constriction, mucus secretion, pain, and itching. Type 2 hypersensitivities involve interactions of antibodies and surface antigens of cells, followed by complement-assisted lysis of these cells. A typical example is mismatched blood transfusions. There are 4 different blood groups, based on types of antigens on the surfaces of the red blood cells. People who are type A have A antigens, and those who are type B have B antigens on the surfaces of their red blood cells. Persons who are type AB have both A and B antigens, and those who are type O have neither A nor B antigens. The serum of people with type A blood contains antibodies against type B antigens, and the serum of people with type B blood contains antibodies against type A antigens. Type AB serum contains neither antibody, and O serum contains antibodies against both A and B antigens. If blood from a person who is type B is transfused into a person who is type A, antibodies present in the type A blood react with the surface antigens on the incoming red blood cells. This leads to complement fixation and lysis of these cells. If blood from a person who is type A is transfused into a person who is type B, antibodies present in the type B blood react with the surface antigens on the incoming red blood cells. This also leads to complement fixation and cell lysis. Persons who are type O lack A and B antigens on the surfaces of their red blood cells and are therefore universal donors. Persons with type AB blood lack antibodies against A or B antigens and are therefore universal recipients. Immune complexes consist of antigens and antibodies bound together. Although large immune complexes are engulfed by phagocytes and removed, intermediate-sized immune complexes are not readily removed. These intermediate complexes react with and activate complement. For example, complement activation in blood vessels causes basophils to degranulate, which causes vasodilation. Immune complexes entrapped in blood vessels or other tissues, such as kidney glomeruli, activate complement, which attracts neutrophils and causes them to degranulate. Neutrophils release enzymes responsible for tissue damage. Harmful effects produced by the mechanisms of cell-mediated immunity are referred to as delayed hypersensitivity. Delayed hypersensitivity is mediated by Th1 cells, also called TD cells, and is responsible for contact dermatitis, tissue damage in a variety of infectious diseases, and rejection of tissue grafts. A contact hypersensitivity, such as poison oak dermatitis, is initiated by contact with the antigen, which is also called the allergen. During contact, a small molecule from the plant, called a hapten, binds to a carrier protein in the host. The carrier protein with bound hapten is ingested by a macrophage, and the hapten peptide is then presented on the surface of the macrophage on a class II MHC. Th1 cells with T-cell receptors capable of recognizing the hapten peptide antigen interact with the presented antigen, become activated, and increase in numbers. When the host comes in contact with the allergen a second time, the Th1 cells react with the hapten peptide being presented on the macrophage and release cytokines, resulting in attraction of more macrophages, followed by inflammation and skin lesions. Characteristic skin lesions appear after 24 hours, reaching their peak at 48 to 72 hours after the second exposure to the plant.
So in autoimmunity, what happens is that the immune system turns and attacks the body's own tissues. And the reasons for this are a little bit unclear. The results are that the tissues are destroyed or are severely impaired. Examples of autoimmune diseases include Graves' disease and type 2 diabetes, type 1 diabetes, rheumatoid arthritis, lupus erythematosus. There's many more. Okay. Why this happens? Some theories include mutations in the immune system or in the target tissue. Other theories include cells that are in anergy waking up and becoming active and attacking healthy tissue. And then another theory is that you're exposed to an antigen that's similar but not identical to your own, that you develop lymphocytes to attack, and they cross-react with your body's own tissues. And of course, it could be a combination of all of these. Okay. Unfortunately, the only way to treat this is anti-inflammatories and immune suppressants, and that makes the individual more susceptible to disease. Okay. Organ rejection is where the immune system attacks a transplanted organ or tissue because the antigens aren't similar enough to the recipients for the immune system to accept it. And the result is that the transplant is destroyed. So the rule of thumb is that you have to have an 85% or better similarity in antigen display in order for organ or a tissue transplant to have a significant likelihood of working. Okay, and that usually comes from a living related donor. Okay, or luck of the draw, where you happen, if you're on the donor list, to match somebody that's recently deceased, and that transplant will end up alleviating the disease, or in some cases saving your life. The immune system is an interesting and multifaceted mechanism that keeps us healthy most of the time. We've touched on some of the most important components. There's a lot of complex ideas in this chapter, and if you have any questions, please contact me by email, and I'll get back to you within 24 hours. I appreciate you listening, and I'll see you in the next podcast.